Welcome back. Our next speaker for this morning is Professor Jens Nielsen, Professor of Chemical and Biological Engineering at Chalmers University of Technology in Sweden. Professor Nielsen is a world-class expert on systems biology and microbial biotechnology, working at the interface of engineering and biology. He currently heads the Systems and Synthetic Biology program at Chalmers University and has founded several companies that have raised more than 20 million euros in venture capital. Today, Professor Nielsen will talk to us about systems biology of metabolism, including the impact on human health and industrial biotechnology. May we now invite Professor Nielsen on stage, please.
Yeah, thank you very much. Thank you to Bertil Andersson and Bengt Nordin for inviting me to talk here. I have a background as chemical engineer, but quite early on I got involved into biology, and that has been a fantastic experience of having this interface as an engineering and actually looking into living systems, and I'll come back to that in my talk. Maybe I could also just say initially here that I actually didn't think that I was going to go into science and engineering as a professor and researcher. I always thought that I was going to do a career in business, but I was really getting inspired during my education, and that basically led as one thing to the other, and then you just get drawn in by the enthusiasm and also all the big questions there are. So in my lecture, I'm going to talk about systems biology of metabolism. There are going to be certain elements that are pretty detailed, but I'll try to an overall level. But basically, my point are twofold, that by studying and using engineering tools for that, which is where the systems biology comes into the picture, we can basically address two of the grand challenges of the world, namely about solving issues about energy supply, as well as also addressing some of key health issues, namely metabolic-related diseases, which is a very large class and very large group of diseases. So first, very briefly about metabolism. Metabolism fuels cells with energy and building blocks. And here I'm going to exemplify it with a yeast cell that basically takes sugar and converts it very efficiently into ethanol. And this has been exploited for thousands of years for making beer, bread, and wine. And this is one of these things where yeast has been involved for and is very efficient in doing that. When yeast is doing that, it produces ATP, which is basically capturing some of that energy that is present in the sugar and in the conversion to ethanol. And also it divides, so it needs to make more protein, lipids, DNA, and RNA that are constituents of the cell. And these components here of the cell are also coming from intermediates in this conversion of sugar to the ethanol. So you can say that this conversion here actually serves two purposes. To generate the free energy as well as the building blocks for this. And this free energy captured here is actually used in this biosynthetic process here. So this is a typical scenario you have in a living cell, that it's converting, it has some catabolic processes that are converting the sugar to some products, as well as it's capturing that energy generated there to build more cells. So it involves many more reactions than I've illustrated here. This is really a rough simplification. And one important point also, that each reaction here is catalyzed by an enzyme. So if we look at that a little bit broader and go into the central dogma, where we have a gene is transcribed to proteins via messenger RNA. And in the context of metabolism, these proteins here, many of them serve as enzymes that are catalyzed in these conversions processes I illustrated before. And when we go on a broad scale at a genome level, we begin to come to these so-called omes, the genome, of course, the transcriptome, the proteome, and we also define the metabolome, which is the collection of all metabolites in the cells. And we can also talk about the quantification of the conversion of these metabolites to each other, which we call the fluxome. So how can we then begin to capture all these thousands of reactions in the cell? Well, we do that by converting the biochemistry into math. And let me give a very simple example of how we can do that. So let's look at a few of the key reactions that are involved in the catabolism of glucose. We have glucose converted into glucose 6-phosphate, further converted to fructose 6-phosphate, and into fructose 1,6-bisphosphate, and then it basically converts further on there. I also included another reaction here in the so-called pentose phosphate pathway, and we can begin to write the stoichiometry of these individual reactions in a very standard way. We do that such that we have the conversion, the consumption of compounds with a negative stoichiometric coefficient, so we collect everything on the left-hand side and we only have zeros on the right-hand side. We then have these stoichiometric reactions here, and we can then put that into a matrix now where we compile for each of the columns representative the stoichiometric coefficient for each of that metabolite, all the different reactions we're looking into. So here we have, for example, for glucose, only consumed in the first reaction. It's not involved in the 3 other reactions, so therefore it's 0, and so on. So now we have a way of actually taking biochemical information and putting it into a matrix format. Now, metabolism is, of course, very large, involving many thousands of reactions. So this is what we are facing with. Sorry, the projection doesn't come out very well. But this is from the classical Boehringer-Mannheim chart of biochemistry of the cell that you probably have seen many times. Now, but what we can do is that we can convert this and go now to this genome scale level. So that's where I started. We say, okay, now let's try to capture metabolism at the genome level. And so basically what we do is that we do exactly as I illustrated before. We take each of these reactions here, the stoichiometry, convert that into these matrices here. But each reaction we have linked via an enzyme to a gene in the chromosome. So you can begin to see that we can correlate each reaction to a gene in the genome. And what is listed here is so-called GEMS, genome scale metabolic models for yeast. And just to give you some numbers about the scale of this, so we have in these models linked, this is from a recent one that we have just put together, about 1,000 genes collected. There are 6,000 genes in yeast, so that means that we are covering about 1/6 of the genes in these models. We have about close to a little bit about 850 unique metabolites, and we have about close to 1,000 unique reactions in this model here. So you can understand that this is a very comprehensive thing that we can only do using mathematics and computers to basically capture. Now, one important lesson that was really eye-opening for me when we started to do this is how connected metabolism is. We learn in biochemistry about how metabolism is organized into individual pathways. But when we started to look at the connectivity between all these different reactions in the cell, it was really surprising. And I'm going to illustrate that with you in a very simple example. Let's take here, again, a few reactions in the cell. The first reaction converting glucose into fructose 1,6-bisphosphate. In the first reaction, we're using ATP, converting that to ADP. And we're also doing that in the third reaction. So these two reactions are actually connected because besides being actually separated here by one reaction, they are both using ATP. And so if you convert this into a graph, and here is a bipartite graph because we have both the metabolites and the enzymes there, you can see that these two enzymes, hexokinase 2 and PFK1, here actually gets connected because they're sharing the same metabolite, namely ATP. We can also take away the metabolite and just have an enzyme interaction graph, but let's just focus on this one here. This is how yeast metabolism looks like when we do this. It's extremely connected, and that is, of course, because sharing of these, for example, ATP, which is participating in 15% of all reactions in yeast. 15 out of these about 1,000 reactions. Another cofactor that is gluing this together is NADH, but also many other metabolites that are mentioned that are used for building biomass, building more cells. They are also really making this metabolism really connected. And so you can see again also, we cannot study these pathways individually. We have to do it in the whole context. So this is where systems biology comes into play. Now, so what is systems biology? Now, you can have an engineering perspective of that and say that systems biology is about quantitative description and also prediction of biological systems. And so you could say that is the analogy with what you've done in other engineering fields, where you try to predict and design engineering or features by completely computerized design, basically design how all these parts are going to be put together to create a functional car. And in terms of biology, we basically want to do the same thing, but here, of course, there is the question, because we do not know what all the components are doing in the cell. We are still lacking information about how some components are actually contributing to cellular function. So therefore, in systems biology, this becomes a twofold approach. It becomes an approach for using in design, like in engineering, but it's also become a scientific approach to begin to understand and get new knowledge about how some of these different components that we don't know what are doing are actually contributing to cellular function. Maybe just one important point here also is because I predict that biology in the future will really be a quantitative science and very much integrated with mathematics. And this is very much an analogy with a really major breakthrough in chemical engineering came when math was beginning to be integrated and you could start to design complete refinery processes and so on, including the detailed description and design of distillation columns. Now, when we talk about systems biology, there are basically two approaches. One is coming from, we can say, the genomics revolution. This is this high-throughput biology. We generate a lot of data using different approaches here, and we then begin to get an understanding about how that system functions by using this combination of math and biology. Or the other approach, which is a bottom-up approach, where we basically put small components together in a quantitative fashion and see how that system actually emerges as a dynamic interaction system. Now, these two approaches here can actually meet in the case of these genome-scale metabolic models that I mentioned, because they are actually bottom-up built, because we take biochemical information, put it into the model, but as we have a link with the genes of each of these reactions, we can use these data and overlay on these models here. So this is illustrated a little bit here. We have the genome-scale metabolic models. We can use them both for model simulation as well as network-dependent analysis. And what is very important here is that because we use the connectivity, the information about that these different genes, about 1,000 genes in yeast, are connected through metabolism. They function in a context. We can use that context in order to drive analysis of high-throughput data. And we have several examples that illustrated we can hereby get new biological information out of that. I'm not going to go into details about that. It's described in some of the papers here. So let me go to the first point, namely metabolism and industrial biotechnology. I'm going to do that relatively short and go on then to biomedicine. So first about industrial biotechnology, the energy and chemical industry is really seeking sustainable production of fuels and chemicals. There's a lot of talk about using biorefineries. So basically having as a feedstock biomass, going through a so-called fermentation process where you have a conversion going on. So this can be, for example, in the classical ethanol process, sugar to ethanol by yeast, and generating fuels or chemicals. And there are already many success stories and many more are surely to come. So in this process here, you have, in this fermentation process, you have this cell factory. This can be yeast that is developed to actually do a process. And traditionally, the industry, the biotech industry, was taking organisms, microorganisms that already did a certain conversion process. For example, yeast is very good to convert glucose to ethanol. Now, ethanol is not always the desired product, so maybe you want to have other types of products out here, so you want to engineer your cell factory for that purpose. Now, here comes a very important point about biorefineries, because you can see the infrastructure can be exactly the same. We can just plug and play this yeast cell factory in here, so we can do engineering of the yeast cell factory and put that into the same infrastructure. So it's a little bit like what the chemical industry has been done with catalyst. But the point is just here, the catalyst we are playing with is much more flexible and have a much broader range of capabilities in converting the raw material into the products of interest. Now, what are some of the problems here? Well, as I said, there have already been some success stories, but the problem have been that going from what we call a proof of principle strain to a strain that can be used in industry, that means it has very high yield titers and productivity, it takes a long time. Many man-years have to be going into that, and also it takes a long time. That's an expensive process. So what we need to do is to develop novel technologies, enabling technologies that can speed up this process and basically move forward faster the implementation of biorefineries. And that's where synthetic biology and metabolic engineering comes into the picture, because here we can begin to use these models I talked about, combined with also advances in synthetic biology to basically construct these cell factories. And what we do is that we take genetic information from other organisms, for example, from plants or bacteria, we can put that into yeast, and we can make yeast produce new products. And we can make a proof of principle first, and then, as I said, we have to do further improvement in order to get the productivity high up. And this is an example here where it's shown that we use this whole network concept through the use of these models. So in my group, we work basically on engineering yeast using these tools from synthetic biology and systems biology to produce a whole range of different products here. And you can see they range from biofuels to commodity chemicals that can go into plastic format, for example, fine chemicals, and also even protein drugs. So this just shows the point that I said, that yeast is really a flexible catalyst here, that we can produce all these different products in the same organisms. And that is, of course, also, you can say, somewhat of a scientific hypothesis here. Can we actually engineer, is yeast metabolism so flexible that we efficiently can produce all these different products? I'm just going to give one very brief example. That's production of sesquiterpenes. This is a class of chemicals that are used very widely. They are particularly produced much by plants, and they go into a number of different products that you all know about. For example, perfumes, both fine fragrances, but also all the kinds of perfumery type products. And they are traditionally produced by chemical synthesis, but this is costly, inefficient, and may not also always give the pure terpenes that you're interested in. So there's a much interest in the industry to go to microbial-based production because it's scalable and so on. You can, of course, extract it from plants, but some of these plants are very rare, and it's difficult to get sufficient quantities of the product that you are interested in. So what we have therefore done is that we have taken genetic materials, genes from plants, put them into yeast, and then yeast can begin to produce these different products here that goes into the perfume industry. And so that is one step, that's the proof of principle, but then we also had to do a lot of engineering of the pathway that leads to the precursor for this product of interest here. So here we use this modeling-based approach and identify different genes that we basically had to express at a higher level in yeast in order to efficiently convert the sugar that converted to this compound up here, acetyl-CoA, and then further into the product of interest. And using this approach here, we could then basically bring up from a proof of principle strain about tenfold the production of this sesquiterpene, and we are beginning to reach now levels where this yeast strain can be taken for industrial implementation, and we can basically replace and have a sustainable production of these chemicals. Now then let me turn to the other point, namely metabolism and human health. So metabolism is involved, of course, in all individual cells. They are absolutely essential for our function. And we have many different types of cells in the human body. It's estimated something around 300 different human cell types. Some of these different cell types are assembled into organs. And can we take this concept of modeling the individual cell types and begin to have a whole organ modeling of metabolism? So that's basically what we are beginning to address in this project that we're calling the Human Metabolic Atlas. And so some of our focus is on liver and skeletal muscle and also the gut microbiome that I'll come to at the end. So we developed an algorithm, because here it gets a little bit more complicated because there's not that much information specifically on the individual cell types. But we can have developed an algorithm where we can generate first a general human network that is basically compilation of all enzymes that exist in human cells. and then take data from different databases where they have looked into specific, for example, is a certain gene transcribed in liver cells, in hepatocytes, or has from another important source in Sweden, Human Protein Atlas, where they have mapped antibody staining of proteins in different cell types and use this information to actually make specific networks. So this is basically the idea we wanted to propose here. So we developed this algorithm, generated 69 different cell types, and then one of the cell types that came out is a liver model, a model for hepatocytes. The good thing about that is that we could take that and compare that with a manually constructed model. Liver has been studied extensively, and so we could basically begin to compare how is our computer-generated model comparing with this manual curation. And it turns out that it's pretty good. There are some advantages, there are some things that falls out, but most of the things that fall out we can actually explain also by the differences in input, so we can basically improve the algorithm for the future. So far so good. The algorithm seems to work, and we get something that is in the right range of metabolism that is functioning in the different cell types. Now then, let's look at the 69 different human cell types that we generated. If we do a comparison of these models, maybe one thing that is quite surprising is that they are very similar in metabolism. We think about all the different cells in the human body doing really many different functions. But actually, all of them share more than 10% of the reactions of a core. And if you expand that and look into, for example, 5 or 10 human cell types, that number will increase dramatically. And actually, only 4% of the reactions are unique to one single cell type. So there is really small sets of reactions that differentiates the cells from each other. And this is maybe a very important point for our understanding. Now, it doesn't mean, though, that metabolism is exactly the same, because what we are not capturing here is actually the actual activity of these different pathways. We are just having like an inventory of these, and that, of course, is the next step that one would like to study. How much are these different pathways used in different conditions, for example? So we did another thing also, because we also used this algorithm we developed, the INIT algorithm, to calculate or identify metabolism of 16 types of cancer. These are cancers in different types of the body, so they're really different. And we wanted to address and see if there's something consistent, different in a normal functional cell and a cancer cell. There's a lot of studies that shows that one type of cancer has one type of metabolism and so on, but it's something that is common for all types of cancers. And basically what cancer cells are doing is that they are beginning to proliferate and divide rapidly. So it's a little bit like the yeast cells I showed you before. So they take a lot of glucose and amino acids that are present in the blood and convert that into lactic acid, but also degradation products of the amino acids, which are amino acetone, with the objective to generate ATP needed for the cell division, as I showed. Now, when we do this comparison between 16 types of cancer and 69 human cell types, normal functioning human cell types, comes out this metabolism. I'm sorry that's projecting really bad here. But what we clearly see is that actually this amino acetone come from amino acid degradation has to be dealt with. These are toxic compounds, so the cell further converts them. That results in peroxide, hydrogen peroxide, which is another very toxic compound for the cell. So it develops a scavenging system here involving biliverdin and bilirubin. So this is something that comes up specifically in the cancer cells. It also develops a route to take care of the methylglyoxal, which is also another toxic compound, and basically degrade that, and that involves glutathione. So these are unique reactions that are characterized for any type of cancer development, any type of cancer of the 16 types that we evaluated here, and this points to new targets for cancer therapy and drug development, clearly. So this is basically, you can say, the vision here. We are building this human metabolic atlas, which will be a computerized version of how the metabolism is in human cells. We can then take clinical data and basically begin to do analysis, visualization, but also identify biomarkers and also use it in drug development. And so we predict that this can be a really interactive database and modeling infrastructure that clinicians and researchers can use directly on patient data. So let me come back and end here with the last example to talk a little bit about the gut microbiome, because there I think it will be some eye-opening information for you. So first of all, we of course think of a human body as the human cells, as I just talked about. But actually, each of us has 10 times more bacteria growing on us than we have human cells. Some people even talk about that we are a walking petri dish for bacteria. Okay, but particularly many of these bacteria are living in the gut ecosystem, where they are of course playing a very important role in degradation of also food, and thereby actually you have an interaction between the gut bacteria living there, the food intake, as well as also actually our own genome probably influences in some way that we don't know yet what gut bacteria actually colonize there. And so there are three terms that are important to define: the microbiome, which is the collection of all bacteria that we study here; the metagenome, this is the collection of all the genes of the microbiome; and metagenomics is basically study of the metagenome. And so what we have been doing is that we are taking fecal samples from people, so we basically get a collection of the microbial community that they have living in the gut system. We extract DNA, perform genome sequencing, annotate functions, and begin to do biological interpretation. Now, this gives us a lot of data, so we had to develop a bioinformatics pipeline for handling that. We call that MEDUSA. So this is metagenomic data utilization and analysis. So we crunch these very large amounts of data. Craig Venter was probably alluding to it on his sequencing talk, or probably also one of the other speakers. And so we can crunch these data and begin to get some biological meaning. And I'm only going to talk about one example here, but we have actually several examples of this, and that is in cardiovascular disease and atherosclerosis. So this is, of course, a major health problem in the world, particularly associated with obesity, but also high blood pressure. And in one study from our close collaborator in Gothenburg, Frederick Beckett and his group, they found actually that in atherosclerotic plaques there are small containments of bacterial DNA. So, and they are most likely derived from the mouth and the gut. So this actually points to that there could be some correlation between the gut microbiome as well as development of atherosclerosis. So we took this pipeline, analyzed from about 30 subjects. Half of them had an atherosclerotic event, and the other ones were healthy subjects. And we sequenced this data here, and the first kind of analysis that came out is that we found that around the 30 people, they are grouped into three categories, actually into three enterotypes, so-called. This is a definition or a term that has been introduced by the group of Per Borg in EMBL in Germany. And so basically what you see is that all of us seems to distribute into three enterotypes. So basically you can now begin to ask yourself which enterotype you have, because also what is interesting is that there is actually an enrichment of the controls in enterotype 1, whereas actually the patients have somewhat enrichment in enterotype 3. So already here we can see that the collection of bacteria you have in the gut may be correlated to whether you are likely to develop atherosclerosis or not. This is further seen in this slide here, which you cannot really see. I'm very sorry about that, but maybe you can briefly see the yellow dots here. The yellow dots here are the patients, and over here we have the controls. And so what we did here, we took all these data and did a so-called principal component analysis to see if there are some features that separates, that are consistent with the controls and the patients. And one thing that was very surprising was that we found that actually two of the controls here were lying together with the patients over here. That was a little bit shocking, and we actually told the clinicians about that, and it turned out that these two subjects, these two healthy controls, actually had an atherosclerotic event after the samples had been taken. And when we take them out, we have a very clear separation of this, showing a very clear and strong predictive power of that. So how does it link to the chemistry of the reactions? Well, we also found a clear correlation between the gut bacteria and the lipid chemistry in the plasma. So there is a clear correlation also there. We also found actually that there is an enrichment of the GS-GOGAT systems in patients. And the GS-GOGAT is maybe interesting because this is an amino acid biosynthesis system that seems to be particularly related to what kind of diet you have. If you have a large vegetarian type diet, you may get enrichment of this versus a beef type diet. So this brings me to my conclusion here. I hope that I've shown to you that science and engineering is really important and key drivers to address some of these really grand challenges of the future: sustainable production of energy and materials, and also ensuring lifelong health through finding ways of understanding how metabolic diseases are affecting us. And so let me thank many of my students over time. This has been, I would say, absolutely essential for my career also, the fact that I have had a chance and opportunity to work with so many enthusiasm-driven and smart young people, and so really many thanks to them. I'm not going to mention specifically who has contributed to this, but this has, of course, as you can imagine, involved a lot of people. Thank you.